Infectious diseases are diseases caused by microorganisms like bacteria, fungi, viruses, protozoa. In this video, we will review vocabulary terms that will help you to describe disease and talk about its causes. Your objectives are to distinguish between the terms primary pathogen and opportunistic pathogen, distinguish between infection and disease, correctly use terms like pathogenesis, etiology, and pathogenicity, explain Koch's postulates and their relevance to disease, describe infectious diseases by distinguishing between how easily they are spread and what signs and symptoms accompany them, and describe infectious diseases in terms of their severity, duration, extent of host involvement, and occurrence. Finally, you should be able to explain the stages of disease. After spending some time in microbiology, it's hard not to have a bit of hypochondria or to not be a germaphobe, but I'm here to tell you that that's not necessary. The truth is that most microorganisms that we come in contact with on a normal day will not harm us. They don't make us sick at all. This slide will hopefully explain why. Let's look at the flowchart on the right side of this slide. When we first come in contact with a potential pathogen, there's a few things that can happen. One thing that can happen is that nothing happens. We get rid of the microorganism. We wash it off, brush it off. It can't survive. Our immune system gets rid of it. It's just gone. Another thing that can happen is that the microbe does attach to some surface of our body and may or may not grow. In the case of transient microbiota, the microbe attaches to some surface of our body, but it doesn't really grow, or at least not usually. Then it can be eliminated either by washing or rubbing or some other mechanism. Sometimes these potential pathogens do become part of our body. They actually attach to some surface and they start to grow there. And most times those organisms will have a commensal existence with us. They're not going to really harm us. They derive benefit and we're mostly unaffected. In rare cases, however, these commensal organisms, because maybe our immune systems are weak, we're under stress, and our defenses are low, maybe the skin, which is one of our largest defenses, maybe it's been damaged, like by burns, and we're susceptible to infection. By one of these reasons, the bacterium or other pathogen actually starts to infect us. Infection is simply a colonization of the body by pathogens. And I want you to remember this part. The ability of a pathogen to cause an infection is due to a number of factors, including entry. So how does the pathogen get in? That portal of entry is key. If the portal of entry is too far away from the tissue that a microbe infects is located, then that microbe is not going to make it to that tissue and infection is not going to happen. The tissue affected is important. The number of pathogens entering is important. There are a lot of defenses that microbe has to get past in order to colonize our body. If there aren't enough of them, then no infection is going to start. And then adhesion. Almost every microorganism has to be able to attach to some receptor on one of our tissues in order to start multiplying. If a pathogen can't find its receptor in our body and it can't attach, then it can't reproduce and we won't get an infection. Now, so that's what happens with microbes that actually start to live and grow inside of us. They're commensal with us. Some microbes don't go that route. They cross the skin and go into some sort of sterile tissue like the blood or muscle, and then they'll start an infection that way. Once an infection starts, there's a couple of things that can happen. One is that the immune system rallies and basically eliminates that pathogen. So it's no longer in our body. If that doesn't happen, then disease results. Disease is an abnormal state in which the body is not functioning right. In other words, it's a change from the state of good health. That's what disease is. Disease can result from an infection, or sometimes it doesn't. Think about diabetes, for example, or heart disease. They don't usually have a microbe involved. So disease is simply an abnormal state in which the body's not functioning normally. Once a disease begins, there's a few things that can happen. The immune system is still fighting the pathogen. It might overcome the pathogen and eliminate it, or the pathogen overcomes the immune system and it lives in our bodies a bit longer than intended, in which case a chronic disease happens. And in the worst case scenario, the human body can't overcome the pathogen and the damage that it's causing, and death results. When we're talking about disease, there are some other terms that are typically used. The first is pathology. Pathology is the study of the cause and effect of disease. You've probably first heard this term when a relative had a biopsy done and the removed tissue was sent to a pathology department for examination. Abnormal tissues from a biopsy may indicate the presence of diseases like cancer or a blood disorder. For infectious diseases, an examination of tissues may reveal the presence of a microbe or the effects of its presence in what was healthy tissue. So maybe the tissue, for example, is damaged. Etiology is the study of the cause of a disease. In microbiology, scientists try to determine what the microbe was that causes a specific disease. For example, human papillomavirus is responsible for 90% of cervical cancer cases, and hepatitis B virus is responsible for 60% of liver cancers. Hepatitis B and human papillomavirus are etiological agents. A scientist or medical doctor interested in pathology is usually interested in pathogenesis as well. Pathogenesis is the development of a disease. So someone in pathology may follow what are the steps that lead to an infectious disease. How does that disease develop? When we consider microbes as etiological agents, we usually also consider pathogenicity. Pathogenicity is the ability of a microorganism to enter a host and cause disease. The more pathogenic a microbe is, the easier it is for it to enter a host and cause damage or disease. We use two terms to describe pathogenicity. Virulence is the degree to which a microbe can infect or damage a host organism. And there are two types of virulent microbes that I'm expecting you to know. The first is called a primary pathogen. These are the probable causes of disease. They cause disease in healthy individuals as well as those who may be immunocompromised or have some other predisposing factor to disease. An example would be Salmonella species of bacteria. For pretty much anyone who ingests these bacteria and get them into the intestines, they will cause diarrhea. Second type of virulent microorganism would be your opportunistic pathogens. Now these are typically less virulent, much less virulent than your primary pathogens. Your opportunistic pathogens may be agents of disease when the defense mechanisms of a potential host are compromised. So for example, E. coli for most people doesn't cause disease. However, in people who are predisposed to urinary tract infections and they get a catheter placed, E. coli may be more likely to cause disease. It's opportunistic. There are certain conditions, like these predisposing factors, that make a particular person more likely to experience disease from that microbe. S. marcescens is another example. It causes pneumonia and urinary tract infections in people who are immunocompromised. So this means that for most healthy people, being around S. marcescens is not a problem. But for people who are immunocompromised and they come in contact with this bacterium in the wrong way, they may be more likely to get a disease or to become diseased. Avirulent refers to microbes that are not regarded as disease-causing agents. These are organisms that rarely or never cause disease in humans. For example, there is a bacterium called Lactobacillus acidophilus that rarely causes disease, if ever. In fact, this bacterium is included in some of the probiotic yogurts that are out on the market today. If microorganisms can cause disease, how do you prove which microbes cause which diseases? The answer is Koch's postulates. Koch's postulates show the direct relationship between a disease and a specific microorganism. The postulates are named for the German physician who developed them. They are a set of steps that microbiologists and epidemiologists use to establish the etiology of infectious disease. The procedure begins when microorganisms are isolated from a group of diseased or dead animals who all show the same signs of disease. The microbes are then grown in pure culture, isolated, and studied so that they can be identified later. Then a small specimen from the pure culture is injected into a lab animal, and the animal is observed for signs of disease. If the animal gets sick and dies, the microbes from it are isolated again. Then they are studied using the same tests as the initial microbe. If the same microbes are found in the dead animal, as in the original dead animals, then it's assumed that the microbe caused the disease and later death of the lab animal. The four major points of this procedure are: 1. The same pathogen must be present in every case of the disease. 2. The pathogen must be isolated from the diseased host and grown in pure culture. 3. The pathogen from the pure culture must cause the disease when it is inoculated into a healthy, susceptible lab animal. And 4. The pathogen must be isolated from the inoculated animal and must be shown to be the original microbe or pathogen. Koch used these steps to demonstrate that Bacillus anthracis causes anthrax in animals, and these steps are still widely used today. There are some instances when Koch's postulates cannot be used. One such example is when multiple pathogens cause the same disease. For example, nephritis, inflammation of the kidneys, can be caused by a number of viruses, including your hepatitis viruses. It's also caused by a number of bacteria like Streptococcus pyogenes, Streptococcus aureus and epidermidis. Salmonella bacteria can cause nephritis, as can E. coli and others. Koch's postulates also are less helpful when some pathogens cause multiple diseases. For example, Streptococcus pyogenes can cause sore throats, but it can also sometimes cause scarlet fever, and then there are skin infections as well. One of the requirements of Koch's postulates is that you isolate your pathogens in pure culture. Well, some pathogens can't be cultivated on artificial media, which means that you cannot isolate them and make them into pure culture. These pathogens have to be grown inside of other organisms. So, for example, Mycobacterium leprae, the bacterium that causes leprosy, is sometimes grown in armadillos in the laboratory because it actually can survive inside of those organisms much better than it can on artificial media. Some diseases have poorly defined etiologies. Initially, we don't know what's causing those diseases. And so you can't really use Koch's postulates because you have no starting basis to do your study. And then some diseases aren't caused by pathogens at all. Can you think of diseases that are caused not by microorganisms but by some other condition in the body? When humans suffer from disease as a result of an untreated infection, there are a number of ways that we can describe that disease. Over the next few slides, I will present some of them. To start off, diseases can be described in terms of signs and symptoms. A symptom is a change in body function that is felt in a patient as a result of a disease. So this is something that the patient feels and has to tell you about. Maybe they feel achiness in their joints, or they have a headache. Maybe they just feel extremely fatigued, or they have malaise. These are all things that you can't necessarily observe when you look at someone. They have to tell you about them. Next, you have signs. A sign is a change in the body that can be measured or observed as a result of disease. So this would be temperature changes that you could measure with a thermometer, or blood pressure changes that you measure with a sphygmomanometer. Or it could be that you notice there's a swelling or redness on the body. Maybe you're measuring heartbeat and you notice that it's slightly fast. You have tachycardia, and you notice that with a stethoscope. All of these are things that you can measure and observe, so they would be signs. A syndrome is a specific group of signs and symptoms that always accompany a disease. So an example of this would be toxic shock syndrome. This is basically a staph infection. It's caused by Staphylococcus aureus, which is a common bacterium on the skin. And in some cases, these bacteria produce a toxin, which inside of the body causes a lot of really negative effects. For example, people may have a sudden onset of high fever. And this high fever will be at least 102 degrees Fahrenheit. They may experience vomiting, diarrhea, dizziness because of low blood pressure, maybe muscle aches or confusion. There's a typically watery diarrhea, sore throat, and then within 24 hours a very characteristic or telltale rash that looks a bit like sunburn that shows up on the skin. Now this syndrome is mostly associated with women who are menstruating, and is mostly associated with tampon use, but I do want you to be aware that men can also suffer from this, as can children and women wearing pads, because it's not the tampon that's causing the problem, it's the toxins made by bacteria on or in the body that are causing the problem. In more than 30% of cases, there's no obvious bacterial infection. It's really the toxins. Some people have actually acquired this syndrome after surgery. Another way that we can talk about infectious diseases is we can describe how easily they are transmitted from one person to another. Any disease that is transmitted directly or indirectly from one host to another is called a communicable disease. So chickenpox, genital warts, tuberculosis, COVID, influenza, they are all communicable diseases. Contagious diseases are diseases that are especially easily spread from one host to another, like COVID, influenza, pertussis, this is whooping cough, measles, and others. Non-communicable diseases are diseases that are not transmitted from one host to another. These are diseases that you pick up because of your interactions with your specific environment. For example, you step on a rusty nail and you pick up a tetanus infection. Or maybe you eat food at a buffet and the food isn't kept up to temperature and you get food poisoning. Or maybe your defenses are low. You're under stress and your immune system isn't working as efficiently. So some of the microbes in your normal microbiota begin to grow out of control and you get sick because of that. That would All of these would be examples of non-communicable diseases. We can also talk about diseases in terms of how long they last. So acute diseases are ones in which the symptoms of disease, the signs of disease develop rapidly, usually within a few days. The signs and symptoms after that initial development can be very intense and severe, but typically these illnesses only last a few weeks, maybe 3 or 4 at the most. So your colds, strep throat, appendicitis, pneumonia, those would be bronchitis, I think I said influenza, I'm not sure, influenza, all of those would be acute diseases. Chronic diseases are ones in which the signs and symptoms develop slowly. So the disease develops slowly, usually over several months. Typically, there's more damage because of how long these particular diseases last. So you can think of cervical cancer due to human papillomavirus as being an example of a chronic disease. Lyme disease that's untreated would be a chronic disease. Hepatitis can be a chronic disease. Subacute diseases are ones that have symptoms between acute and chronic timetables. So if we say chronic diseases last around 6 months or so, or sometimes longer, then subacute is going to last between 1 month and about 6 months, potentially longer. So if you think about someone who has a respiratory illness and they have a lingering cough that goes on for a month or two after they've actually recovered from the illness, that would be a subacute condition. Endocarditis, this inflammation of the heart, it would be another example of a subacute disease. Latent diseases are ones in which there's a period of no symptoms. So someone gets sick initially, that would be like an acute phase. Then the microorganism that caused that illness hides out in the body and it's inactive or dormant for a time. Then, usually due to stress or some other factor, that microorganism may become reactivated and cause another disease later on. So latent tuberculosis would be an example of this. Varicella zoster, which causes chickenpox initially, but later causes shingles, is known to go through periods of dormancy, but it can be reactivated later. We can also describe diseases in terms of how much of the body is affected by the disease. So a local infection, for example, is one in which the bacteria that are colonizing the body, causing the infection, they're located in one specific area. So for example, this is a MRSA infection, but you can see that the bacteria, as kind of indicated by the redness here, they're located to just this area. They're not extending beyond that area in either direction. So that's a local infection. Systemic infections would be more like chickenpox over here on the right, where multiple parts of the body are being affected. So with chickenpox, it's not just the skin that's affected, but also the nervous system. And we're going to see this infection all throughout the body. People have had lesions inside their mouths and on the bottoms of their feet and on their hands, just everywhere. So that's a systemic infection. A focal infection is one in which you have a focus of infection. So you have something like this, but the bacteria or their toxins spread from that local infection and affect the rest of the body. So for example, endocarditis. This is inflammation of the endothelial surfaces of the heart and valves. It can be caused when commensal organisms in the oral cavity are disrupted. So common dental procedures can cause the bacteria to actually enter the blood and be in the blood. That's called bacteremia. Being in the blood provides a pathway for those oral bacteria to make their way to the heart and cause inflammation. So that's a focal infection. We start out in one location, but the microbes and their toxins spread to other locations and cause disease. On the last slide, I mentioned bacteremia. Bacteremia is simply the presence of bacteria in the blood. It can arise from a serious infection in the body or from something as harmless as vigorous toothbrushing. Most of the time, only a few bacteria are present in the blood, and they can be eliminated pretty easily by the immune system. People usually don't even know that they've had this condition. Septicemia, on the other hand, is much more serious. Here you have bacteria in the blood, but they're originating from a serious bacterial infection somewhere else in the body, like the lungs. The bacteria from the lungs go into the bloodstream, and we get symptoms like fever, rapid heart rate, rapid breathing rate, increases in white blood cell count. Bacteremia and septicemia can trigger sepsis, which is a serious body-wide inflammatory response. It also can lead to fever, rapid heart rate, rapid breathing rate, increase in white blood cell count, but it can also lead to heart failure, lung failure, and kidney failure. Sepsis can also cause septic shock, which is a dangerously low drop in blood pressure, which causes organs to receive too little blood and they'll start to malfunction or fail. Septicemia, sepsis, and septic shock are all life-threatening conditions. Many bacteria have the ability to produce and secrete toxins. When these toxins wind up in the blood, toxemia results. The symptoms of toxemia kind of depend upon what the toxin is that's been released. Viremia is the presence of viruses in the blood. Another method of talking about disease and infection is to talk about primary infections versus secondary infections. Primary infections are those that cause an initial illness. Secondary infections are opportunistic infections that occur after a primary predisposing infection. Let me give you an example of what I mean. In 2009, Americans were worried about an influenza strain called swine flu. The strain number was H1N1. During an influenza infection, usually it's the very old and the very young who are most susceptible to complications of the flu. And unfortunately, they are also the ones who are most susceptible to dying from the flu. During this outbreak, however, young adults were dying. These victims were infected with H1N1 and they eventually overcame it, but the inflammation and damage caused by the various white blood cells fighting the infection was so severe that the victims were predisposed to a secondary bacterial infection that led to pneumonia and ultimately killed them. H1N1 was the primary infection that made the conditions ripe for the growth of the bacteria which caused the secondary infection. Predisposing factors, like the lung damage that I mentioned on the last slide, make the body more susceptible to disease. Some examples of predisposing factors include short urethras in females, which make females more susceptible to urinary tract infections, inherited traits like the cystic fibrosis transmembrane conductance regulator. This is a protein that controls the movement of ions across cell membranes, and when it's not present or when it's mutated significantly, it leads to the production of mucus in the lungs and around the vital organs of cystic fibrosis sufferers, and this can make them predisposed to lung infections. Climate and weather can make us more susceptible. For example, it may favor the growth of mosquitoes, which carry malaria or dengue fever. Or maybe it's cold and keeps everybody inside in close quarters where they can pass disease more readily. Fatigue. Fatigue leads to reductions in immune function, so you're less able to fight off infections should pathogens enter your body and try to grow. Age. We know that the immune system is not as efficient the older we get. It's also not very efficient when we're very young, but the reasons are different. When we're very young, the immune system just hasn't seen enough pathogens to adequately protect us from infection. And when we're very old, it's just less efficient because we're old and the proteins don't work as well. And there's a number of different factors at play there. Lifestyle can make us predisposed to infection. Perhaps someone is an intravenous drug user and they um, are using risky behavior that may bring them in contact with blood-borne pathogens. Or maybe someone is being unsafe sexually and they predispose themselves to infection that way. Could be that someone is receiving chemotherapy treatments and their immune system is weakened as a result. That can predispose people to disease. Diseases can also be described in terms of stages. You've probably heard of the incubation period before. This is the time between when a microbe enters a potential host and the appearance of the first symptoms. You might remember that this is when, if we're talking about influenza, the influenza comes into the nasal passages and lodges on the epithelial tissues at the back of the throat. There it starts reproducing and triggers the first of your immune responses. And it's those responses that lead to the appearance of your first symptoms. The incubation period is followed by the prodromal period. During the prodromal period, you get indistinct kind of general symptoms. This is when you might feel bad, but you don't really know exactly what's going on. Maybe you experience headache or nausea. Maybe you have muscle aches and you just feel really fatigued. Between the prodromal period and the acme or climax period, your signs and symptoms usually increase. At the acme or climax, that's when they're going to be at their greatest. So this is when we usually see the appearance of rashes, high fever, chills, dry skin, pale complexion. This is when you can look at someone and tell that they're really sick. As the immune system begins to fight the microorganism and symptoms start to subside, hosts will enter a period of decline. So here, again, signs and symptoms are subsiding. You might notice sweating, fewer coughs and sneezes, fever is gone away, and there's a gradual return to normal skin color. The period of decline is followed by the period of convalescence. This is the return to a state of good health. How long do each of these stages take? It depends upon the disease. For most of your acute diseases, recall that someone will travel through all of these stages within a couple of weeks to a month. This slide correlates the number of microbes in the body with the stages of disease. You'll notice that over here during the incubation period, the number of microbes is really low. Remember, this is where you first come in contact with the pathogen. It attaches to one of your tissues and it starts to grow. So we're going to see a slight increase in the number of microbes. That's followed by the prodromal period. Here, the symptoms or signs are starting to intensify a bit. This is when you start feeling sick. You get those non-specific cues that illness is on the way. That's followed by the period of illness. So notice we're getting a steady increase, exponential increase in the number of microbes. Now, what's happening with the immune system at this point? Well, There are only a few cells in your body that can recognize the antigens on the surface of a pathogen in the absence of a vaccine. So it takes time for those specific cells to actually find the pathogen that's multiplying in your body. And it turns out that the pathogen is much more efficient, much faster at reproducing than the immune system is at actually locating that source of infection. Once the immune system is able to recognize the antigens on that microorganism and start fighting that microorganism, we'll start to see a leveling off on the growth of the pathogen, whatever it is, and eventually a decline. Usually it takes about a week for the immune system to find a pathogen and start fighting. Once the immune system starts fighting, the number of microbes goes down, the signs and symptoms of disease also start to decrease. Eventually, we'll have such a great reduction in the number of microbes that will enter the period of convalescence. This is the return to a state of good health. This is when you really start feeling better and you feel like you've turned the corner on the infection. The last way that we want to describe disease is in terms of its occurrence, how often it happens. Two words that you need to be familiar with are incidence and prevalence. Incidence is the fraction of a population that contracts a disease during a specific time period. That's the number of new cases. So for example, we might say that the number of new cases, the incidence of new cases of COVID-19 from March 5th to April 12th is 2,474. Prevalence is the fraction of a population having a specific disease at a given time. So prevalence would do new cases as well as old cases. So we would expect that number to be a bit higher. Sporadic diseases are diseases that occur occasionally in a population. So they occur rarely and without regularity. There may be a single case or maybe just a few scattered cases. So food poisoning cases, typhoid cases, or bacterial meningitis cases would be an example of sporadic disease. We can also talk about endemic disease. So endemic disease is disease that's constantly present in a population. These are the diseases that are always around. So for example, here in the United States would be things like Lyme disease or salmonella or meningococcal disease, chickenpox, influenza or colds, histoplasmosis. These are all diseases that are constantly here in the United States. They're endemic. Epidemic diseases are diseases that are acquired by many hosts in a given area in a short period of time. The rate at which a disease or an epidemic spreads is influenced by the number of people involved and what kind of immunity they have. Now let me give a plug for herd immunity. Herd immunity is when you have immunity in the population, most of the population of a given area. And herd immunity is conferred by vaccination. So the idea is if you have a large population of people and they're all vaccinated against the same disease, for example, influenza, then that means that the number of cases of influenza in that population should be few to zero because there aren't enough suitable hosts to transmit the disease from one person to another. The importance of herd immunity is that it protects the more vulnerable people in that population. So it protects young kids who can't get vaccinated and protected against disease. It protects people who maybe have egg allergies and can't receive a vaccination because of that. Or it protects people who are immunocompromised. It protects people who are elderly and and maybe can't receive a vaccination. So kind of the same way that wildebeests and other large animals herd, so they exist in these large groups and they arrange themselves so that the very young are usually protected on the inside of the herd. Herd immunity tries to do the same thing. We've seen the effect of a breakdown in herd immunity here in Massachusetts, also down in New York, where we have populations of people who are not immunizing against measles. And as a result, they've started to see outbreaks of measles in these two locations. Pandemics are basically worldwide epidemics. At the time of this recording, we're in the middle of the COVID-19 pandemic, so you're living in history. You can see exactly what it is to be in the midst of a pandemic just by looking around you. So this was a whopper of a lecture, but I really wanted to make sure that all of the disease terminology was together for you in one video. In this particular video, I distinguished between primary pathogens and opportunistic pathogens. Remember, the big difference between them is that primary pathogens can make a host sick, whether they are healthy or not. Opportunistic pathogens usually make weakened hosts sick. So people who have lowered resistance to disease because of stress, maybe it's medications that they're on, they just can't fight the disease, fight pathogens as easily. We distinguish between infection and disease. Remember, infection is simply colonization by a microbe, and that microbe can be a pathogen. It doesn't necessarily mean that the body is not working properly. Okay? Disease is a change from the state of good health when the body's not working properly. And that may or may not be due to an infectious disease. We use terms like pathogenesis, etiology, and pathogenicity. I explained what Koch's postulates were and their relevance to disease. Remember, this was that series of steps that relates a specific microorganism to specific diseases, a specific disease. We also talked about when you can't use Koch's postulates. We talked about infectious diseases and how you can distinguish them based upon how easily they're spread. So that was contagious versus contagious versus communicable versus non-communicable. We also talked about what signs and symptoms are. We described infectious diseases in terms of their severity, so that was acute, subacute, and chronic. We talked about duration and extent of host involvement. And finally, we talked about the occurrence of disease as well. So that was incidence, prevalence, and we talked about endemic, pandemic, epidemic. We talked a little bit about herd immunity as well. Finally, I explained the stages or periods of disease. All of this is fair game for your exam, so you want to be familiar with these words and what they mean. My next video is going to be on disease transmission. I look forward to seeing you there.